The adapter hypothesis is used to describe a tRNA molecule because it uses the information provided by the messenger RNA, which is a codon sequence, and complementary binds that with an anticodon, and then the associated amino acid is then released from the tRNA and makes the amino acid chain or a protein. When you look at the definition of translation or even the word transfer from tRNA, you think of turning one kind of information into another in order to communicate or transmit that information. This is really similar to the definition of an adapter, a device that connects two things that cannot be connected directly in order to transmit information or signal. When I thought of that, I thought of when you try to play a song from your phone onto a speaker and you need an adapter. So in this case, if you would like to play a song on a speaker or express a gene, the original message, which would be the DNA from the phone, is the chosen song, which is the mRNA. Then the cable port, which is represented as a codon, matches with the adapter here, or the anticodon. This then, as a result, transmits the signal to the auxiliary cord, making an amino acid chain. The auxiliary cord then sends the protein, or signal in this case, from the phone to the speaker where it's played aloud. So then the protein would be expressed as a gene. Something that can inhibit this is antibiotics. For example, the antibiotic streptomycin, it changes the conformation of the bottom part of the ribosome in translation to where it'll read the messenger RNA incorrectly and match it with the incorrect transfer RNA molecule. So instead of isoleucine, a ribosome complex infected by streptomycin might read it as threonine. This could result in non-functioning or incorrect proteins or cell death in bacteria. Another example is the antibiotic tetracycline. In bacteria, its action is to block the connection of the transfer RNA to the ribosome mRNA complex, which prohibits translation and protein synthesis altogether and will cause cell death. Another example would be the antibiotic chloramphenicol, and this just prevents the peptide bond from forming between adjacent amino acids, allowing it not to make a chain and causing no proteins to be produced and causing the bacteria or cell to die.